Chapter 6, Section 2, focuses on the second part of photosynthesis, which is the Calvin cycle, which was named after Melvin Calvin, who is actually an American scientist who did a lot of work on the pathway details. And the Calvin cycle is where the plants use their energy that had been stored in ATP and NADPH during light reactions to produce their food, or organic compounds, or sugars. So this part does not depend on light, which is why it's also called the dark reactions. It's also called carbon fixation, meaning that we're taking carbon atoms from CO2, or carbon dioxide, which is one of the reactants of photosynthesis, and bonding them into sugars. So this process has a number of steps. It begins when CO2 diffuses into the stroma, which is the liquid portion of the chloroplast, and combines with a molecule called RuBP, or RuBP. When it does that, it splits that molecule into two 3-carbon compounds. So we say we're fixing C3 plants with this process. Those new compounds that were made are called PGA. Those PGAs are then converted into G3P, which in some older textbooks are also referred to as PGAL. And they're converted into those molecules with the help of some of the stored energy from the light reactions, the ATP and NADPH. Now at this point, one of those G3P molecules is going to leave the cycle to make the food energy, the carbohydrates. The other G3P gets converted back to RuBP and restarts the cycle so that you're constantly able to begin again with this cycle and it doesn't have to stop. So here's a diagram of that. You've got carbon dioxide flowing into the stroma. Okay, we're combining that with RuBP. Okay, that's this big molecule here. These are the carbons and some phosphate groups on the end. And that essentially splits that molecule into two 3-carbon compounds. Okay, we add in a little bit of energy from ATP and NADPH, and those molecules get converted into PGAL, or G3P. Okay. One of them will go to make the organic compounds, and the other one will get converted back into RuBP with a little more ATP, and we can start the cycle again. So you can fix a carbon dioxide with each turn of this cycle. Now the stomata are an important part of the plant. It's a small pore that's on the underside of the leaves, and that's what's going to allow gas exchange. So that's what's going to be able to bring in CO2 into the plant and release the oxygen that is made there. Now the problem with that is that water can also get lost through those open stomatas. So when stomata are partially closed, they can help to decrease how much water they lose, but if they close too much, then they're not going to be able to bring in enough carbon dioxide, which is going to slow down this carbon fixation process. Here's another diagram. This is a cross-section of a leaf. Here's one of those little pores, and you can just see that the gas exchange can occur through here, but you could also have water being lost through those pores. Now, not all plants follow this carbon cycle. Most of them will fix their carbon through that process. They're C3 plants, but there are a couple other possibilities. C4 pathways and CAM pathways. With a C4 plant, the carbon is going to be fixed into a 4-carbon compound instead of a 3-carbon compound. So if you remember at the beginning of that cycle, we took the RuBP and the carbon dioxide and split them into two 3-carbon compounds. Well, with this, we're making a 4-carbon compound. Plants that use this generally will partially close their stomata during the hottest part of the day, and that will help to decrease some of their water loss. So some plants that will fix their carbon this way are corn, sugarcane, crabgrass, some other different types of weeds can also do that. And then CAM plants are going to be in even hotter, drier climates than the C4 plants, and they will operate at night. So they actually will close their stomata during the daytime when it's going to be very hot, so that they don't lose very much water, and then they'll open their stomata at night so that gas exchange can continue to occur. So things like cacti and pineapples, they operate through this CAM pathway. The last thing I want to talk about is the rate of photosynthesis. Now, the rate of photosynthesis can be affected by three major factors: light intensity, carbon dioxide levels, and temperature. So this diagram here represents both light intensity and CO2 would follow this same pattern. This is our rate of photosynthesis, and this is our light intensity. You could also say this is our CO2 levels. Now, if you increase how much light a plant is given, you're also going to speed up how fast it can photosynthesize. So that's where we're getting this increase here. But eventually, it will be going as fast as it possibly can go, and it won't be able to bring in any more light to photosynthesize faster. Same thing happens with the carbon dioxide. The more of a reactant that you have, the faster the reaction can go. So it's going to keep speeding up until eventually it's going as fast as it can process, and it'll level off. Now, temperature behaves a little bit differently. It's more of a bell curve. You're going to have a high point here at the average range. So with your rate of photosynthesis, if the temperature increases, so will photosynthesis. It will speed up. But eventually it's going to get too hot for the plant to operate at its most efficient. And some of the enzymes that help in this process will start to break down in that extreme temperature. So at that point, the rate of photosynthesis would start to decrease again. So at temperature, it will increase, but eventually it will decrease again if it gets too hot. With light intensity and carbon dioxide levels, photosynthesis increases and then will eventually level off.